Thank you all for having me here today. I will be here today and until lunch tomorrow. And I'm very happy to discuss things if you think what I present is sufficiently interesting after. I'm going to talk a bit about the research I've done at the Swedish University of Agricultural Sciences, my PhD, but also a research proposal that I have been submitting last week, and which would entail some collaboration between me and SLU and Domi, and I want to use your compost facilities. Just shortly about me, I'm now doing a postdoc in the ecosystem ecology lab at the Swedish University of Agricultural Sciences. My PhD and my research so far has been on soil microbial diversity and functioning. Like I said, I'm writing a grant application now, which is on the effects of organic amendments on soil microbes and soil microbial functioning. Some cute photos of my PhD supervisors, which were Mohammed Bahram. He has now moved on to become a professor in Denmark, and Janne Bengtsson in the middle, he has retired, so now it's only Maria left. My thesis is found at the Swedish University of Agricultural Sciences, if you're interested. The presentation structure now is, in the first part, I'll just present what I did in some of the main results from my PhD. It's quite microbial ecology focused, so it's big ecosystems and a lot of quite natural systems as well. And then the second part, I will talk about the research project proposal. And then finally, some questions. So my PhD thesis was on land use change, impact on soil microbiomes. Right now, I'm doing a postdoc until next year, looking at plant soil dynamics across the same systems that I researched in my PhD, which I will present now. And I'm doing some follow-up experiments to look more at targeted at some of the findings I found from the field. I'm going to start by giving you a brief history of land use changes, because that was my main topic, land use changes in Sweden, that is, but it's actually quite representative for the large-scale land use changes that have occurred in Europe and actually the global north over the past 300 years or so. And I'll start by showing you, this is like a digitized map. In Sweden, we're blessed with very precise and meticulous bureaucrats that go and map things very carefully. And they have done that for quite some time. So in the early 1800s, even back to the 1600s, they were going out in the landscape and mapping exactly what is this field, who is living here, and so on. And they were doing this for tax purposes, to get as much tax as possible. But they were saving these really nice land use history maps, which researchers now have been digitizing, and then you can see changes in the landscape over centuries of land use change. So essentially what you see is a quite representative landscape from central south Sweden, which consists pretty much of 75–80% grasslands, and the rest is crop fields, and the little gray dot there is where people were living. That's a farm. I'm going to show you what the landscape looked like 100 years later, same area, early 1900s. Quite similar. What has happened is that the crop fields have expanded on grasslands, and that is due to the invention of crop rotations and the introduction of leguminous herbs into the crop rotation, so that you would fixate nitrogen through clover and other leguminous herbs, and then fertilize the soil. So the other parts that were typically too poor fertility-wise to grow crops on could now be cultivated for crops. Another interesting thing is that the crops were always located either on the most fertile patches in the landscape or really close to the house, because that's where you could bring your manure. Otherwise, it would be too heavy to transport your manure. So this is really a big land use change in the 100 years there, because the ecosystems, the landscapes in Sweden were really stable from medieval age up until the early 1900s. Very few like large-scale changes, because the fertility of the soil dictated the use of the landscape. The reason we have so much grassland as well is due to it gets quite, you don't know that because you went in summer, but it gets quite cold in winter. All the animals need to stay for 6-7 months inside, and you need to feed them with hay. The hay you cut in summer. So a lot of the landscape was actually not, it was grazed or used for hay cutting to bring the feeding for summer, for winter, sorry. Okay. Now in the 1950s, what has happened is mechanization of agriculture, tractors, but also fertilizers, obviously, mineral fertilizers. What we see is a massive change from what it had looked like pretty much since the medieval times. And all these grasslands that used to be cut or grazed has now turned into forest. Why? Because with the fertilizers, we no longer need the area that we needed before to get the grass. So it's a representation of the intensification of agriculture that happened. And when we talk about intensification, we usually talk about like yields, how much we get out and so on. But I find this really impressive in the sense that you can see the effects on a landscape level. This is just a little landscape, but imagine this all across Sweden and across most parts of Europe. The land use changes that has occurred here are at an almost unimaginable scale. We also have patches here that used to be croplands that are now, the brown is abandoned. So when you stop grazing and cutting, the grasses start growing more, and then after a while, it enters into succession. After a while, you have essentially trees growing, and it slowly turns into a forest. And then this is a continuing trend now. So I find that when I was doing my PhD, I traveled across the whole of Sweden. It was beautiful in the sense that I got to see a lot of things. I went from the far south to the far north to sample grasslands across different stages of abandonment. And you really see this is still ongoing. Like there's so many grasslands that used to be managed that are abandoned, no longer managed, and they're slowly turning into forests. But essentially, we used to have a grassland-dominated super regional landscape that has now turned into a heavily forest-dominated landscape. So the afforestation that has occurred in 150 years, it's essentially not even two generations of forestry. So a forest cycle usually lasts 100 years from when it starts growing until you cut them, and then it starts regrowing again. That's how much the landscape has changed. So this is a large-scale afforestation that has happened historically, that is still happening. Abandonment and afforestation of open land, grasslands. There were typically quite a lot of trees in the grasslands, but they were open because they were managed. When abandonment happens, you stop managing, the grasslands enter into secondary succession and become forests. And the end point, this is really important, the end point, because succession, the literature or the theory of succession in ecology is, when there's a fire in a forest, it's a cleared patch that then starts regrowing, grasses, taller grasses, herbs, nettles, and so on, and then shrubs, bushes, and after a while you have a forest. And the early ecology, the field of ecology, almost starts with the study of succession. And there's a very influential idea that says that the end stage of succession is a mature climax forest, which is an ecosystem in balance. This is the idea of the balance of nature and so on, that you have a spontaneous progress that ends with a climax, a stable ecosystem. We don't have that in this. It's really important to stress, because when people talk about succession, you imagine very often that the forest that you end up with is a climax forest, natural. All the forests, 80% of them that have grown on what was previously open land, end up in production forest systems. In Sweden, that is typically monocultures and clear cutting. Clear cutting is you take a patch of 2 hectares, you cut every single tree, you leave 5 or 10 trees, and you plant again with the same species and get it to regrow. That's really different from a natural climate forest. This process, abandonment and overgrowth, succession of open land and production forestry in Sweden, are the 2 main drivers of biodiversity loss. In the Red List species guide, they've identified about 1,500 threatened species to each process. It's like beyond comparison the strongest drivers of biodiversity loss in Sweden. Also in Europe, I don't have the same numbers, but the trends are similar in Europe. So we have really, really contrasting pictures here, which is typically ecology. It depends where you are, when you are there, or so on. When people talk about afforestation, it's and deforestation, that's really in the global south, where the forest cover is of natural forest are quite high. Deforestation there is typically a really bad process, like for biodiversity. In the global north, it's a bit the opposite, because the landscapes have been open and kept open, so that the biodiversity over time has adapted into this high biodiversity hotspot landscapes with open management. When you have a abandonment of that and a growth of forests, it becomes actually a big threat to all the habitats of the biodiversity adapted to open land. Big drivers of above ground biodiversity loss. We don't really know the impacts when it comes to soil microbial, not on a large level. We know from local patches and so on. So that was my thesis topic, was to look what's going on with the soil microbial communities in this process. So I went from the south of Sweden all the way up of the Arctic Circle, and see it's really similar. You see a lot of grasslands that are being abandoned and growing into succession. I paired every grassland site I had with a forest site that was really close geographically, so that I could compare what the grassland is going to look like when it eventually becomes a forest and is incorporated into this forestry practices. The grasslands were in differing stages of secondary succession then. I collected soil samples from this, and then I sequenced them with amplicon, metabarcoding, and shotgun metagenomics to get an idea of how the taxonomic biodiversity is affected, but also the functional diversity. And this is a sketch of a secondary succession managed grassland that starts abandonment, and then they gradually grow into forest. So these are the successional categories that I looked at. And just to illustrate what they typically looked like, a very interesting aspect of this changing landscape and changing demographic processes over the last 200 years. As a result, we have more horses in Sweden today than we have cows. And we don't have like farms are centralized, concentrated into really like high intensified farms. The small scale farms of 20 to 30 to 50 cows are pretty much gone. That's why the abandonment occurs. But where it's still managed a lot of times, it's by horses. Early successional grasslands could typically look a bit like this. You have like a quite drastic change in the vegetation where these taller grasses and there's quite a bit of nettles and stuff, they start taking over, and then over here you start see tree line starts encroaching and taking over. Late successional grassland could look like this, even taller. Sometimes the grasses would be up to here. I'm quite tall, but the grasses are even. And then you see that here spruce. So in Sweden, spruce and pine are the dominating tree species. So evergreen, and here the spruce are like. It's really a fascinating process. They like send out these pioneers that come, and then goes quite fast, like 20, 30 years. It's really like they some come and then the rest come after. And after a while, when you have a forest, thick forest of spruce, it's like the impression is so different from a grassland. It's really dark. Being under this canopy, it's so dark. Fully a forested site. So this is actually quite a nice forest because it's not monoculture. There's some different tree ages and things, but generally you would have more homogeneous. So it's even aged forests and typically one or maybe two species. Lots of blueberries. Right. So for the results. When it comes to taxonomic diversity, this is fungal diversity, bacterial diversity, forest, late successional, early successional, and managed grasslands. The dashed line here is the baseline, which I set as the forest sites. When they have become like the reference forest sites, the diversity is going to be here. And what we see is higher fungal and bacterial diversity, especially bacterial diversity in grasslands than in forests. So there's essentially a loss of taxonomic microbial diversity in this land use change process. So that corresponds quite well with the above ground picture we have from like butterflies and plants and things, that you have a loss of biodiversity in general when you go from open grasslands to forests. And then when it comes to genetic diversity, so here I've focused especially on caseines, which is like the microbial genetic set to metabolize or degrade carbon substrates, produce enzymes that degrade carbon substrates. But I'm only showing caseines here, but I did the same for nitrogen and phosphorus genes. Interestingly, this is what it looks like here. So for fungi, the diversity of carbon genes, so the genetic repertoire they have to degrade carbon substrates, it's much higher in forests. And for bacteria, it's actually similar. These are a bit higher in grasslands, but it's not significant. So overall, yeah, an increase in the genetic diversity. And this is true for carbon, also for phosphorus, not for nitrogen then, but there's an increase or stable genetic diversity. And what this means is that we have an increase for both fungi and bacteria, an increase of genetic relative to taxonomic diversity with afforestation. And in ecology and you know the biodiversity depletion, but also the concept of ecosystem stability, there's a one point that is really coming up all the time, which is of why do we need to care about biodiversity at all, especially when it comes to bacteria. There's hundreds of thousands or more, and in my plots there's typically 15,000 species in a single soil sample. If we lose some, what's the problem? So when you have a lot of taxa that perform similar functions, you've got a high functional redundancy. So they keep functioning and the ecosystem processes keep working, even though you lose biodiversity. If you have, like in forest, few taxa for many functions, you have a high functional specialization. They're very specialized, but their redundancy is quite low. So if you have, you lose biodiversity for some reason, drought, fires, global warming, there's a higher risk that your ecosystem functions stop or they decrease. And I took this trend of redundancy and specialization and put it into a framework which has been developed by an Italian researcher quite recently, where you plot an axis or you analyze the axis of functional redundancy versus specialization. And this is what it looks like when I analyze my sites. This is for bacterial taxonomic diversity versus carbon cycling genetic diversity. You have a specialization axis that goes here with the forest sites, and then a redundancy axis that clusters a lot with the grassland sites. So overall, the pattern is that of high taxonomic diversity over here in managed grasslands, high functional redundancy. The more succession happens and the more afforestation, the microbes get fewer taxonomically and more functionally diverse. So the specialization of the microbes increases, but the redundancy decreases. And that's pretty much the main conclusions of my PhD. There are some other parts too, but I just sum it up by saying that afforestation is a driver of biodiversity loss in Europe. Majority of grasslands become production forests for microbes. There's this relative increase of functional or genetic diversity, but a trade-off, it seems like, between specialization and redundancy. And the implications of this in real-world systems is what I'm going to look a bit more at in my postdoc now, and do an experiment of essentially taking grassland and forest sites in microcosms and artificially sterilize them, and then put back a gradient of diversity so that you simulate biodiversity loss and see how much the functions, the ecosystem functions, are impacted by that. But generally in Sweden, at least, and in quite a few European countries, with climate change, there's increasing. There was a very alarming report recently in Sweden that showed that the Swedish forest, the rate of growth has gone down in Swedish forests. It's gone up since the 80s, and now it has started decreasing. And they don't really know why that is, but they suspect it's because of increasing drought that affects the symbiosis between mycorrhizal fungi and the trees, and also make the trees much more susceptible to insect damages and so on. So I think it's going to become more, it's a bit theoretical when you look at microbial diversity because they're so diverse, but the real world implications might start becoming more tangible the more the ecosystems get stressed. Right. So the background for me being here today is that I've been writing on a research project proposal. So this in Sweden we have this early career grants that you can apply to get 4 years of postdoc funding essentially, that covers your salary and some research expenses, and there's a mobility element there where you have to stay abroad. And I would like to go to France. Specifically, I've been writing this with Sébastien Barrault at the Institute of Environmental and Ecology in Paris, and with Laurent Philippot at INRA in Dijon. And the topic here is compost ecology. I want to move a bit away from the big natural systems and look at what happens in managed systems when it comes to microbial functioning, and where you actually apply certain composts directly, and how does that affect the function. So it's related to my thesis topic in the sense that still about microbial functioning, but it's in much more controlled ecosystems. The background here is that there's a lot of food waste in the EU. 55% of that comes from households. A lot of it goes into landfills, methane problems. At the same time, there's a lot. They say right now that soils is a sexy topic, and that's because they found out that they have managed to quantify the extent of soil erosion and degradation across the EU. They say 62% of soils are degraded. That's referring to specifically to erosion, but there's other really important degradation aspects of soils, like loss of soil fertility and biodiversity. And a lot of this stems from the separation here between compost being generated and processed separately, agricultural soils being fertilized with mineral fertilizers, but there's very little reapplication of organic material, which creates the fertility, the structure, and the biodiversity normally in agricultural soils. So a lot of the erosion and the degradation comes from the lack of recycling between the organic waste products and the extraction that occurs in agriculture. So with this project, I want to look at 2 different conversion pathways through which organic waste products are put into organic amendments. The main topic is effect of organic amendments on soil microbial functioning. So digestate and aerobic composting, which are then subsequently applied to soils in a cropping system. And here, the microbes in the soil that are already in the agricultural soil, they do some really important and fundamental functions that are of high importance in this cropping system. The nutrient cycling is the maybe main one, but then carbon sequestration by transforming the digestates or the amendments that are applied, they are being microbially transformed into particulate and mineral associated organic matter, and then stays in the soils for longer or shorter time periods depending on the transformation and the amendment type. So it's essentially a way of looking at the carbon sequestration potential and the fertility of the soils in relation to microbes. And then there's here the disease suppression part, where a lot of soils have a natural high disease suppression capacity, which means that when you introduce pathogens there, they often get killed off by the residing microbes through antagonistic interactions. And increasing the disease suppression capacity also means that you would be able to reduce the fungicide or the pesticide applications. So that's another highly desirable microbial function in these systems. The aim is to assess long-term effects of repeated organic amendment applications on soil microbial function in cropping systems. 3 work packages. First is to do a general mapping of what happens to the soil microbial functioning, like I showed in the results earlier. How does it impact the diversity of these different genes and the overall functional profile of the microbiomes? And here I'm talking about different organic amendments. So whether you use sewage sludge or green household waste, green waste, varying the type of waste products and the conversion pathways to look at the long-term effects of those on this functioning part. Then I want to test and determine the disease suppression capacity of the microbial soils after many applications of organic amendments, and determine the carbon transformation capacity. And to do this, I'm going to make use of 3 existing long-term field trial sites where they have been applying different kind of organic amendments for about 30 years. One in Sweden, one in Denmark, and one in France. In France, it's the Caliagro trial sites outside Paris. And so this is mainly with composting. In these 2, there's digestion, digestate treatments as well. And the first project is just to sequence these sites, take soil samples, and look at the metagenome functional profiles. Second test is a greenhouse experiment to test the disease suppression capacity then. Essentially taking soils from the field sites, growing plants in there, and then inoculating them with fungal pathogens that are important for cropping systems, and see how the natural disease suppression makes some of the plants more damaged than others. And the hypothesis is that certain type of organic amendments promote a higher disease suppression capacity of the microbiomes. Then measure the plant damage, assess disease suppression, and relate to the metagenomic profile. So in this way, you get a more mechanistic or ecological insight into why do certain amendments actually give better disease suppression and better soil health in general. It goes through the microbiome in this aspect at least. The last work package is when you apply the organic amendment, whether it's digestates or composts, to the soil, there's this transformation here, which is really quite magical almost. It's not just microbes, but mainly microbes. And there's, in this stage here, where the fresh organic amendments here, which vary in their composition and residue profile, they get incorporated into the soil and the microbe starts decomposing it. You have this this step is happening here where they start transforming the carbon, and quite a lot of it is respired as carbon dioxide, and the other parts are turned into microbial biomass or into this associated to the mineral matrix of the soil, which creates stable or more particulate forms of organic matter. So these relate to the overall pool of soil organic carbon and tells us a bit of how the expected residence time and the sequestration capacity then of these soils. And to do this, I would use 13C labeled Y, so it's a stable isotope of carbon that I would take mix with the type of composts, food waste products they use in the Caliagro. So for example, with food waste as in here, run it into a compost reactor and derive a 13C labeled compost after this. This compost is applied to the soil just as we saw before, and this whole step here happens. But because you have a known input of 13C in the compost that you apply to the soil, which has a natural background 13C to 12C ratio, you can trace essentially. The 13C is a tracer to see how much of that, when I apply this once, how much of this carbon is actually converted, transformed in these different pools. So we can, by measuring the carbon dioxide heterotrophic respiration of the microbes, we see how much of the carbon that I add is respired in heterotrophic respiration, how much is incorporated into microbial biomass. This one here is a really popular research topic at the moment because there's a strong link between microbial necromass and very recalcitrant organic matter. So the theory is that the more necromass you have, the more stable is the organic carbon, and it's going to resist further degradation. This is very, very difficult to measure though, so I'm going to keep at the overall microbial biomass. And then by fractionation, derive the proportion of particulate versus mineral associated organic matter at the end, and measuring the 13C of these pools. So that gives an idea of how much of the compost you add is respired, is turned into biomass, and is incorporated into the soil in these different pools. So the requirements for this work package is a place where you can continuously measure respiration of labeled carbon. It's labeled material input for the compost. It's compost reactors, the field soils, and the lab for the carbon fractionation. This is why I'm here, because you have absolutely wonderful compost reactors where it's possible to, with labeled carbon plant material, co-compost it with other things and get the labeled compost at the end. I don't know any other places, at least not in Sweden, and as far as I've seen, where you can do this in such a nice way. So that's a bit of my background and the people I've been working with so far, and a bit of what I hopefully will be doing during the coming 4 years or so. I will get the answer to the grant application in November. This is just finishing a painting, a nice painting by a Swedish. He was actually a biologist or so before he started, stopped that and became a painter instead in the 1800s. And it's from the region of around Uppsala where I'm currently studying. And the lovely thing about this era and this period of painting is that they were super realistic. It's oil paintings like super realistic scenarios. And it gives you a good idea about how the landscape was managed and what it looked like at the time. So 1870s, Right now it's just like forests all across here, but it was much more open, fences for animals, graze, quite a lot of trees, and the people and animals that were keeping the landscape open. So, thank you.